Assalamu alaikum. I hope you are well. You are not tired? Are you tired? Thank God.
We are full of
Thank God
energy.
you are full of energy. Okay. Shukran lillah. You are not tired. Today, in this lecture or in this episode, we are going to do HLA typing test. HLA typing test, please. Please. Where are you going, Haji? Take your seat. Are you clear? HLA typing. Why do we do HLA typing?
In a transplant.
It's important for transplantation. Why HLA typing is important, should be done before transplantation?
Because the body rejects the.
It's because recipient's immune system will reject the transplantation. We do HLA typing to prevent transplant rejection. Am I right?
Yes.
Okay. Why do we do HLA typing for prevention of HLA transplant rejection?
To know
What's the relationship?
to know if its immune system accepts the new substance, new kidney or something.
Immune system accept new cells,
Yes.
new tissues,
Yes.
like kidney, organ. No. Yes, that's right. But why from the several or many, so many antigens on the cells of the body? For example, on the kidney, we have several or many antigens. Okay? We have so many antigens on the cells of a kidney, which is going to be transplanted. Why we only check some antigens, like HLA? Why we don't check all of the antigens? We only check some of them. What's the difference between the antigens that we should check and the antigens that we do not check them? Do not check for their similarity. They are not important in transplant rejection or acceptance. What's the difference between two types of antigens? Let me ask my question again. On the kidney, which is going to be transplanted, there are many antigens. Is that clear? But from a huge number of antigens, we only check some of them, including HLA antigens. Why we only check some of the antigens on the transplant for prevention of transplant rejection?
Because the other antigen similar.
Yes,
Human.
most of the antigens are similar in persons of the population, in different persons. But HLAs, I'll tell you again in the next slides, we have different kind of HLA between persons of the population, so we should check the similarity between HLAs of two persons, recipient and donor, in order to prevent transplant rejection. So, first of all, I'll review HLA, human leukocyte antigen, or MHC, two names for one thing. MHC, major histocompatibility complex, another name for HLA, major histocompatibility, compatibility of tissue which is going to be transplanted, complex. And I'll talk about HLA typing or assessment of HLA profiling, and mainly HLA typing, and then I'll talk about, in summary, about biosafety consideration in the medical laboratory. For determination of HLA profile in transplantation, several tests are done, but we only in this lecture are going to talk about HLA typing, not others. So HLA antibody checking or cross-match is done, but we are not going to talk about it. Please have a look here. An APC, a T cell. Antigen presenting cell, T cell. So, tell me what's the function of HLA? The function of HLA in immunology, in immune system. What's the function of HLA? Yesterday you talked about it completely. In group. Please.
Recognition of the self from the pathogen.
One role of HLA is determination of self from non-self. But HLA proteins have very important role in immune activation, in immune system. Do you remember the function of immune?
Representing the T.
Presenting antigen. Presenting of antigen on the APCs and other cells of the body is done by HLA. For example, if I am an APC, I showed you, APC needs a tool to present antigen. This tool is HLA. Do you remember? APCs and all nucleated cells need a tool to present antigen to T cells. Without binding of T cell to MHC, we won't have T cell activation. So T cells should bind to HLA and antigen with their TCR, T cell receptor, in order to become activated. So APCs and other nucleated cells need a tool, MHC, to present antigen to TCR on the T cell. This was first signal. Do you remember? We needed two signals for T cell activation. First signal by TCR binding to MHC antigen, second signal by co-stimulators. Do you remember?
Yes.
Clear everything?
Yes.
Yes.
So, it's a function of HLA immune system. And you know that we have three classes of HLA. HLA class 1, class 2, and class 3. HLA class 1 antigens or proteins are expressed on all nucleated cells of the body. All nucleated cells of the body have HLA class 1 on their membrane, on their surface. All of the cells, nucleated cells. So, MHC class 1 proteins are expressed on the nucleated cells and they present cytosolic antigens or intracellular antigens to cytotoxic T cell or CD8 positive T cells. Do you remember?
Yes.
CD8 positive T cells, in order to become activated, their TCR should bind to HLA class 1 protein, antigens that are in the cleft of HLA class 1. HLA class 1 is only a tool. Antigen is important in T cell activation. It is clear, isn't it?
Yes.
Antigen is important, of course, for T cell activation, but this antigen should be in the cleft of HLA. HLA class 1 is expressed on the surface of all cells of the body, and they present intracellular antigens or cytosolic antigens to CD8 positive or cytotoxic T cells. On the other hand, MHC class 2 proteins or antigens are expressed on the APCs and some other immune cells only, not on the all nucleated cells. So APCs and some other immune cells have HLA class 2 that they present extracellular antigen to T helper 1 or CD4 positive T cells. Is that clear?
Yes.
CD4 positive T cells, extracellular antigen expressed or presented by HLA class 2, which they are on the APCs. CD8 positive T cells or cytotoxic T cells is activated by MHC class 1 plus intracellular antigens which they are on the nucleated cells of the body plus APC. Is that clear?
Yes.
And we have HLA or MHC class 3. We have several genes that all of them are called MHC class 3. They are only called a name HLA class 3. It's because all of the genes of HLA, which is called HLA MHC, major histochromatin complex. It's called complex because we have several genes located on chromosome 6 together, close to each other. So, but several genes are located between the location of HLA class 1 and HLA class 2. So, as they are located between the location of HLA class 1 and 2, so they are called HLA class 3. But they do nothing in HLA antigen presentation. Whenever we are talking about HLA typing, antigen presentation, T-cell activation, APC, and many other immunologic terms, we are talking only about HLA class 1 and 2, not HLA class 3. It's only a name for several genes and their products. That these genes are located between the genes of HLA class 1 and 2. So we do nothing with HLA class 3. We are only talking about HLA class 1 and 2. You can see here the structure of class 1 and class 2 HLA. This is a HLA class 1 and this is a HLA class 2. In HLA class 1 we have an alpha chain and one beta 2 microglobulin. Do you remember?
Yes.
HLA class 1 is composed of two different chains. The first chain is called alpha chain, the second chain is beta 2 microglobulin. But in HLA class 2, we have one alpha and one beta chains. There is a difference between them, you can see here. Besides, the genes of alpha from HLA class 1 and alpha and beta of HLA class 2 are located in HLA region. But gene of beta 2 microglobulin is located on a different chromosome, chromosome 15. All of these complexes are located on short arm of chromosome 6 in human. And for HLA typing, we do not need to type the beta 2 microglobulin. So typing of beta 2 microglobulin is not needed in transplantation. But alpha chain of class 1 and alpha and beta chain of class 2 should be checked before transplantation. But it's not needed to check beta 2 microglobulin type. Question, comments? And as you remember, we have three HLA class 1, HLA A, HLA B, and HLA C. And three class 2 HLAs, HLA DQ, DQ, and DR. As reminding. Do you remember HLA A, HLA B, HLA C as a class 1 HLAs, and HLA DP, DQ, and DR as a class 2?
We didn't take it. We didn't take it like this names.
You haven't heard this?
No.
Complement system?
No, I'm talking about HLA, please.
No, we didn't.
I'm talking about HLA. We have two different classes of HLA.
We know
HLA
the
type
classes.
1.
We know the classes, but like this names we didn't know.
Okay, I'm telling you. We have HLA class 1 and HLA class 2. And in each of them, we have three different HLAs. We have three HLA class 1, HLA A, HLA B, and HLA C. And three HLA class 2, HLA DP, DQ, and DR. They are three different HLAs of class 2. Is that clear?
Yes.
Yes.
And they are located, as I said, on the short arm of chromosome 6 in human. Sorry. Okay, this is chromosome 6, and HLA or MHC genes are located on the short arm of chromosome 6. All of them are here. there is a very important note here, please. We have one gene for HLA class A, one gene for HLA C, sorry, one gene for HLA B. All of them are HLA class 1. So HLA A, HLA B, and HLA C, all of them are expressed on the nucleated cell as they are HLA class 1. All of them present intracellular antigen. All of them present cytosolic antigens. 2 CD8 positive cytotoxic T cells. All of them are HLA class 1. And you can see here that we have 2 genes for DP, 2 genes for DQ, and even more than 2 genes for DR. But for class 1, we have only 1 gene for each one. It's because, as I said, only alpha gene of HLA class 1 is here. Beta 2 microglobulin is located somewhere else. But here, this is HLA-DP alpha gene, HLA-DP beta gene. As I told you, HLA class 2 is composed of 2 chains, alpha and beta. Each one has its own gene. So we have 2 different genes that encode alpha and beta. So it is DP alpha and DP beta, DQ alpha and DQ beta, DR alpha, and even in HLA-DR we have more than one one gene for beta chain. Beta 1, beta 2, and beta 3. Is that clear?
Yes.
Cool. You can see here again HLA-BP, QR, B, CA, HLA class 1, all of them HLA class 1, and all of them HLA class 2. We have some other genes between the HLA class 1 and class 2, which they are called HLA class 3. Because they are located between the genes of HLA. So they do nothing in antigen presentation, as I told you, and transplantation. And again, DP alpha, beta, DM alpha, Q alpha, beta, R alpha, beta 1, beta 2, and sometimes beta 3, 4, and 5. And B only alpha, C only alpha, A only alpha. Is that clear? HLA genes, please. Haji. Here, please. Haji's. Haji.
Old man. Hadis mean old man. Hadi gentleman.
Gentleman.
Continue, stop, please.
Mr. Haji.
Yeah, Mr.
Hajia means for ladies.
Yes, Hadi.
Hajia. Mrs. Hajia. Okay. HLA genes are inherited as haplotype. What does that mean? Please, we do not have enough time. HLA genes are inherited as haplotype. What does haplotype mean?
Set from mother and set from father.
Yes. What does haplotype mean?
Half.
No, half no. A set of genes they are located close to each other and they are inherited together to the child. Okay? And when we say 2 sets, each person has 2 sets of HLA, one set from father, one set from mother. We mean one set is this: HLA A, B, C, DP, DQ, and DR. One set means this, all of the HLAs. And this is indeed one set of HLA genes. 3 HLA class 1, 3 HLA class 2. And another set from mother or father. One of them from father, one of them from mother. Is that clear? Yes. They are inherited together. I have an image for this in next slide. Besides the genes of HLA are inherited codominantly. What does that mean? These concepts are very important. Very, very important. These concepts would be helpful for you in the clinic if you are going to work with tissue transplantation. So, knowing the protocols of the test is not as important as these topics or concepts that I'm trying to explain to you. So, genes of HLA are expressed codominantly. What does that mean? Codominant. It's a biologic term.
What alleles of mother and father pass to the child?
Again, please.
What alleles of mother and father
Yes.
expressed by the child?
All alleles which is inherited from father and mother are expressed simultaneously. Let me give you an example. If father blood group, not HLA, blood group is A, and mother blood group is O, the blood group of the child would be A. The blood, I'm talking about phenotype, not genotype. The blood group would be A. Father A, mother O, or mother A, father O. It doesn't matter. The child would be A. But if father is A, mother is B, the father would be AB. It's because A and B are codominant, have codominant expression, but A is dominant to O. When we have A and O, we have only O antigens on the surface of the cells, not O antigens. It's because A is dominant to O, and B is dominant to O as well. But A and B together, they are codominant. The expression of A won't affect the expression of B. Whenever the expression of one gene won't affect the other one on the other chromosome, we have two genes of two sets of each gene, one from in the chromosome from father, another in the chromosome from mother. So whenever expression of one gene do not affect the expression of the other one, it's called codominant expression. For example, if a person okay.
Doctor, if we have B and O, B and O,
If the mother or father is B, the other one is O, the child would be B.
so always we don't choose O, always choose another one.
I didn't get what you said.
Okay, I understand.
الدمنات
Tell me after the class. I'll try. So, please, for example, if a person have A, B, C, D, P, D, Q, and D, R from one set from father, another set from mother. All of them are expressed on the surface of cells. The expression of A from mother will not affect the expression of A from father. The expression of B from mother will not, besides the expression of A will not affect the expression of B. B will not affect C, and so on. All of them are expressed simultaneously on the surface of cells. Is that clear?
Yes.
It's called codominant expression of the genes. Another important term. This is the basic for HLA typing. This says why we do HLA typing. The genes of HLA are very polymorphic. The most polymorphic gene of the human, and I think other species, are HLA genes. They are the most polymorphic gene of the body. What does that mean? HLA genes are the most polymorphic genes of the body. What does that mean? This concept is very important for all you are doing today in this lecture. I mean. What does polymorphism mean?
Many shapes.
Many shapes of
Structure.
One gene. Different alleles or different shapes of one gene in the population. It means that we do not have only one HLA-A. We have thousands of different HLA-A and thousands of different HLA-B, thousands of HLA different C and so on. But as I told you, beta 2 microglobulin typing is not needed in transplantation. It's because beta 2 microglobulin gene is not polymorphic. Beta 2 microglobulin of all human is same. There is no need to check it because it's completely same. If we injected beta 2 microglobulin of one person to another person, we won't have any immune reaction. It's because they are completely same. Is that clear? But if we transplant a tissue from the donor that its HLA is A2, if a tissue containing HLA A2 is transplanted to a person with HLA A45, so we have two different HLA-A. The HLA-A2 in the body of the recipient will be considered as a non-self antigen, as a foreign. Is that clear? An immune system will become activated and we will have antibody and T cell activated against that, the cells of the tissue. Is that clear?
Yes.
So we have thousands of HLA-A antigens and HLA class 1A, thousands of B, thousands of C, and so on in the population. And the aim of this lecture is to check the HLA typing, is to check the type of HLA on the surface of cells of donor and recipient for tissue transplantation. As you can see here, on the surface of cells, we have 12 different HLAs. 3 class 1 from mother, 3 class 2 from mother. And on the other hand, 3 class 1 from father, 3 class 2 from father again. So we have 6 HLA class 1 from mother and father, and 6 HLA class 2 from mother and father. This is a haplotype. A haplotype which is inherited from, for example, mother. We have 6 different HLA, 3 type 2, 3 type 1, class 1 and class 2. And another haplotype from the father, 3 HLA class 2 and 3 HLA class 1. So on the surface of cells, we have 12, there is this possibility that we have 12 different HLAs, 2A, 2B, 2C, 2DP, 2DQ, 2DR. And we should type all of them. And we are going to check the type of these HLAs on the cells of donor and recipient by doing the HLA typing test. Clear?
Yes.
Question, comment?
No.
No? Alaikumullah, if you have question. خب. As I told you, each person has 2 sets of HLA. Each person has 2 sets of HLA. One set from father, another set from mother. When we say a set from father or mother, we mean one HLA A, B, C, D, B, DQ, and DR, please. And another set from mother. And for the mother, similar to the father. Each person in the society, in the population, has 2 sets of HLA. When we say 2 sets, each set contains 6 genes at least. 6 genes, 3 HLA class 1, 3 HLA class 2. And 6 other from the father. So each person has 12 different HLA class, different class of HLA. And for example, this child will receive A from the father and C from the mother. So when it received HA from the mother, it received one set of HLA, 6 HLA genes, and 6 other from the mother. Is that clear?
Yes.
Please, question, comment, no? And as I told you, this table shows you the polymorphism of HLA. We have more than 2,000 HLA-A in the population, more than 3,000 HLA-B, and so on. As you can see here, for HLA class 1, we have only one gene. But for HLA class 2, we have BP alpha, 22 different BP alpha. BP beta, more than 500, about 600 BP beta 1. Is that clear?
Yes.
When we say a gene is polymorphism, it means we have different shapes, different alleles of that gene in the population or society. It's because we need to check the type of HLA on the cells of tissue, on the cells of recipient and donor, in order to find similar HLA type between the donor and recipient. For example, if I'm a recipient, I need a kidney, my HLA should be typed to find out at what are my HLA genes, and in a group of donors, we should do the HLA typing to find out the similar HLA between donor and recipient. Is that clear?
Yes. Doctor, is it possible to find similarity with someone not from your relative?
Yes. Yes.
Is it a good chance
But there
or
is-
very rare? Is it a good chance?
It's,
or
yes.
very rare?
No, it's rare. It's because most of the patients that are waiting for transplantation, they cannot find a compatible transplant. And before finding a compatible transplant, they die. It's very common. Most of the patients that are waiting for an organ, they couldn't find the compatible organ, and before finding, they might die. Question? Comment? Let me skip this one. So, please go to the laboratory right now, and you should done one step of your test in the laboratory, and then get back to the class again.
Okay. Now?
Yes, for the rest of the lecture. Go to the laboratory, do one step.
Good afternoon, everybody.
Hello.
Today we wanna do the HLA typing in the laboratory. We need fresh blood. We have it. We take blood. We dilute the blood with PBS. What is PBS? It is the isotonic buffer. For dilution, the blood, we need isotonic buffer. We use PBS. We dilute it already, and it is ready to use for you in your rack on your bench. You have another tube that it contains of a special material that we call it 5-hole. 5-hole is a concentrated sugary buffer for separating the lymphocyte from the whole blood. Because of it has a difference of density between the 5-hole and the whole blood. After centrifuge, you can separate the lymphocyte. The 5-hole is so cytotoxic for the cells of the whole blood. Because of that, please pay attention. Don't mix the blood with the 5-hole. I'll show you how you can use the 5-hole for separating plasma without mixing them with each other. First, I push the bubble and I take the blood in this way. After that, I put the tip of my dropper on the surface of inner layer of the tube, and very slowly I push the bubble of the dropper. Now you can see you create 2 layers. The upper one is blood and another one is 5-hole. Do it very slowly and don't mix the blood with 5-hole. and add all of the blood that you have on your bench, in your rack, in your tubes, all of the blood, you must add on the surface of the, do it and you finish it, bring it for us. We put it in the centrifuge and we centrifuge it at 1,800 RPM, 20 minutes. After that, when we will be bring it out from the centrifuge, we will separate the lymphocyte from, I will show you how you can separate the lymphocyte. For now, if you don't have any question and if it is clear, you can go to your bench and do this one. And after that, bring your tubes for us to put it in the centrifuge. Is it clear? Okay.
Come in, please. Okay. Take your seat, please. Hajiye. Take your seat, please. Okay. HLA typing, haplotype inheritance, codominant expression. I'm talking about the important keywords. Haplotype inheritance, codominant expression, and polymorphism in HLA genes. All of them are clear? No question about it?
No.
Okay. No question. For tissue or organ transplantation, the main problem, the main obstacle for graft, tissue graft means transplantation, another term for transplantation. The main problem in tissue transplantation is rejection of the tissue, transplanted tissue I mean, by immune system. It's the main problem of tissue transplantation. And the aim of HLA typing and other typing like blood grouping is to minimize, please, I'm saying to minimize the risk of transplant rejection. We try to minimize the risk of transplant rejection. Always there is a risk for transplant rejection, but we do some tests to minimize the risk. Okay? The main problem is immune system rejection of the transplant, and if we want to have a successful grafting or successful transplantation, first of all, we should try to find compatible tissue or donor. The first factor that is very important in the fate of transplanted tissue is finding a matched or compatible tissue in terms of their antigenic properties, I mean. Between the more compatibility, the less likely of rejection of the transplantation. The less compatibility, the more risk of rejection of the transplantation. Another thing that is very important, recipients who, transplant recipients always should take immune suppressive drugs. It's because, as I told you, HLA genes are the most polymorphic genes of the body. The most polymorphic genes of the body. But it doesn't mean that they are only polymorphic gene of the body. They are the most polymorphic gene. Is that clear?
Yeah.
HLA genes are the most polymorphic genes of the body, but they are not the only polymorphic gene of the body. We have other polymorphic genes that some of them haven't been identified yet. So it's because always there is risk for transplant rejection. And it's because the patient should receive all the time. The transplant recipient should take immune suppressive drugs for
reduce the immunity
to reduce the risk of transplant rejection. Is that clear?
Doctor,
We only
for how much? For the rest of his life?
for the rest of his life, yes. transplant recipient should take immunosuppressive drug for the rest of their life, because it's not possible to find 100% compatibility between recipient and donor.
Yes.
Would that make him more vulnerable to diseases?
Yes.
We try to give them a balanced immunosuppressive drug, but transplant recipients always have more risk of infectious agents. You're right. But we try to make a balance between this and a longer life with immunosuppressive drug and higher risk of infectious diseases. Okay, so first of all, we should try to find a compatible organ between recipient and donor, and it's not possible to find 100% compatibility, so always there is difference between recipient and donor, always there is risk of immune activation, immune reaction against allograft or graft or transplantation. When we say transplant rejection, we mean activation of immune system, for example, production of antibody or T cell activation against the cells of the transplant, and activation of immune system, and then production of antibody or invasion of cytotoxic T cells to the transplant will kill the transplant cells. So transplant rejection means killing of transplant cells by immune system of recipient. It's an immune reaction. Is that clear?
Yes.
And we do HLA typing to minimize this risk, to prevent transplant rejection as much as possible. Question, comment? No? We do except HLA typing and some other tests, of course, blood grouping is done before HLA typing. It's because you know that we have different blood groups in the population, in the person. So a recipient and donor should have similar blood group as well. We are not allowed to transplant, for example, a person with blood group A to a person with blood group O. Besides, we will do HLA typing and other tests. But today we are going to talk about HLA typing. There are several methods to detect the types of HLA. As you know, as I said, we have thousands of HLA A, thousands of HLA B, thousands of C, and hundreds of other. Is that clear? Do you remember? Yes. And by HLA typing, we are going to determine the type of HLA of recipient and donor, if they are compatible or not. And several methods are used to this aim. One of the simplest method is serology method. Serology test. Serology test or serology tests are a group of tests that in all of them we use the reaction of antibody with antigen to detect one of them. Please, serology means using of the reaction of antigen and antibody to find one of them, to check one of them. For example, we want to check if there is HPV inside the blood of one person or not. In the laboratory, we have produced an antibody against HLA-B in the company. HPV. Anti-HPV antibody is produced by the company, and we have it in the laboratory right now. And we use this antibody to check the blood of the person if there is any antigen inside it or not. Reaction of antibody by its specific antigen. All the tests that we use the reaction of antibody and antigens, generally they are called serology tests. So the simplest way to type the HLA of donor and recipient is serology test. So in serology test, we use antibody. You can see in the next slide. The test which is serology test, which is used to type the HLA of donor and recipient in serology method, is called complement-dependent cytotoxicity assay. Complement-dependent cytotoxicity assay. You should remember that whenever an antibody binds to a cell, IgG and IgM, I mean. Do you remember we have different classes of antibody, but two of them can activate complement. After binding of one antibody to a cell, IgG and IgM, I mean, we will have complement activation after that. Is that clear?
Yes.
CDC, do you remember from yesterday's lecture or previous lecture? Complement-dependent cell cytotoxicity was one of the function of humoral immunity or antibody function. Do you remember? So we use this test to determine the type of antigen. After binding of antibody to the cell, we will have activation of complement, then formation of membrane attack complex. Do you remember? After forming of membrane attack complex, what will happen to the cell? Lysis of the cell. Membrane attack complex will make holes on the membrane of the cell. As a result of forming holes on the membrane of the cell will die. Is that clear? We will have cell lysis. So, let's see what happens in HLA typing. Until now, in the laboratory, we took the blood from recipient and donor, for example, and we added the blood on the follicle, whole blood, we added the whole blood on the follicle to separate PBMCs. You know that in the blood we have several types of cells. Neutrophils, eosinophils, monocytes, lymphocytes, red blood cells, platelets. Do you remember? There are different types of cells in this blood, peripheral blood or circulation of the human. Is that clear? So, we took the whole blood or fresh blood from the donor and recipient and we added the blood on the follicle to separate PBMC, peripheral blood mononuclear cells, PBMCs. The mononuclear cells of the blood are mainly lymphocytes and monocytes. So we use the PBMCs of donor and recipient to check the type of their HLA, to determine the type of their HLA. Until now, you only have separated PBMC. In the next step, we will do the rest of the test to type the HLA. Is that clear?
Yes.
Okay. So, in the laboratory that you go, you will have PBMCs that mainly contains lymphocytes. Is clear? So, the question is this. The aim of this test is to check the type of HLAs on the surface of these cells. Okay? For example, we want to check if the person, we are going to check if the person has HLA A02 or not. As an example, we have several A, several B, several C. For example, we want to check if the cells are expressing HLA A02 or not. We want to check this. If we want to check this, we need an antibody in the laboratory, anti-HLA A02 antibody. In the laboratory, we have already now anti-A02 antibody. It is produced in the company and we have it in the laboratory. We will add this antibody to these cells. If these cells are expressing HLA-A02, this antibody will bind to the cells. Is that clear? If the cells do not express the A02, the antibody won't bind to them. Okay?
Yes.
The question is this. if these cells are expressing A02 or not. We check this by adding up its specific antibody. So, we have separated the PBMCs, then we will add the antibody, for example, A02 to the cells. If A02 is expressed on the cells, the antibody will bind to the cells. And then we will add complement source to the test. We will add complement to the test. After adding of complement, if the antibody is bound to the cells, we will have complement activation. Okay? Cell bound antibody on the cell and activation of complements. And then membrane attack complex and making holes or pores on the membrane of the cell. They are real images which are taken by electronic microscope. You can see the holes. All of them are made by membrane attack complex of the complement. So, cells which has or have A02, anti-A02 bind to them. After adding of complement, we have activation of complements and forming of holes or pores on the membrane of the cells. And then we will add a dye, mainly eosine, it's a dye, to the cell. If the cells, after making hole, the cell of course is dead. Dead cells have a lot of pores on it, but live cells do not have any pore on it. Is that clear? So, after adding of dye, if the cells are dyed, indeed, if they have pores on the membrane, the dye can go inside the cells. The cells indeed can absorb the dye, and we call it a stained cell. So a stained cell means antibody binding, complement activation, forming hole, and absorption of dye. And it means that these cells are A02 positive. Is that okay?
Yes.
This means that these cells had A0 positive, that antibody anti-A0 positive bind to them, attached to them, and then we had complement activation, then we had pore or hole formation, then we had dye adsorption in the dead cell. But in the live cell, there is no antibody binding, there is no complement activation, there is no pore or hole formation, there is no dye adsorption. So, if we add antibody to the cells that they contain the corresponding antigen, we will have a stained cell. But if we had the antibody to a population of cells that they do not have the antigen, we won't have a stained cell. All of the cells will remain alive after the test. Is that clear?
Yes.
It is called complement-dependent cytotoxicity assay. So, I said you only one antibody for one antigen, as an example. But I told you that we have several HLAs, so we should check several HLAs. I only explain you one example. One antibody, one antigen. If it was on the cells, we had a stained cell. If it wasn't on the cell, we hadn't a stained cell. Is that okay?
Yes.
But in order to check the presence or absence of several HLA antigen on the cells, we need a test. We need a plate. Go, please. It's too lazy. We have a plate with several wells on it. They are wells. This plate is called multi-well plate. Multi-well plate. In the bottom of each plate, we have one antibody. So, one well for A02, another well, for example, for B02, another well for B25, another well for DR25, and so on. So at the bottom of each well, we have a different antibody. And we will add the PBMCs of donor or recipient to all of the wells simultaneously. It's because we want to check the presence or absence of several HLA antigens. Indeed, we are doing the test I explained you in several wells. All the process I explained are taking place inside the wells, but the antibody is different. Is that clear?
Yes.
Let me write it down here. We have several wells. At the bottom of this well, we have antibody that are quoted in the company. They are attached to the bottom of the wells. Okay, this one for A02, this one for example B25, this one for DP45, this one for DQ for example 10. The test is done inside of the all wells. If in this well we have a stained cell, means binding of antibody, activation of complements, making whole, dye adsorption, so it would be A02 positive. And in another, then we will check the wells under the microscope. I'm checking the wells under the microscope. In the next well, if we do not have binding of antibody to the cell,
It's negative.
we won't have antibody binding, so we won't have complement activation, we won't have pore or hole formation, we won't have dye adsorption. So they are not stained under the microscope. And we say it's B25 negative, and so on. All the wells are checked one by one for recipient and donor separately. And then we will have a pattern of positive and negative, and we will compare them together. If they have same HLA antigens, they are considered compatible. If they have different HLA antigens, they are considered incompatible. We are not allowed to do the transplantation.
Doctor, so we do it for the donor and the receiver?
For recipient and several donors.
Oh, okay.
For I'm the recipient, I need a tissue. So the test should be done on several donors to find one similar donor.
Okay.
Is that clear?
Yes.
Question, comment? Oh. It's too lazy. So, you are working, when you are working on the laboratory, some biosafety considerations should be taken into consideration. It's because you are working with biologic samples, blood, cell, serum, plasma, urine. All of them should be considered contaminated or infected. HPV, HCV, HIV, and some other infectious agents. So you are working with biologic samples, and you should consider them always as contaminated samples. And besides, you are working with dangerous or hazardous chemicals. Some of them are flammable, some of them are corrosive, and so on. So you should take some biosafety measurements to protect yourself, your family, your friends, your surroundings. It's because you have to wear lab coats, gloves, and you shouldn't eat and drink in the laboratory. I'm talking about medical laboratory. You should take care of your personal stuff, like cell phone,